Hi, my name is Caitlin Levine, and this is my Decomposition Lab project. So, for my project, for my control bin and my experimental bin, I used a leaf, a screw, stick, banana, a tortilla chip, paper towel, cardboard, chip bag, and copy paper. For my control bin, I only did 3 sprays of water, and for my experimental bin, I did 3 sprays of water and 3 sprays of white vinegar. For my independent variable is the amount of vinegar added, and my dependent variable is the decomposition of the trash objects that I chose to use. So my hypothesis was if the trash objects are sprayed with vinegar and water, they will decompose faster due to the acidity of vinegar. So these are my week 1 pictures. You can see everything looks pretty normal. The soil was kind of damp when I first added it into the bin, so the soil looks a little darker. There's no coloring on the copy paper. The chip is fresh, the banana is fresh, the leaf still has vibrant colors, and the stick is pretty fresh. I mean, I got this from a dead branch, but it is still a stick. And there's no mold or anything on it. There's no white bumps or any sort of fungi. Same for the experimental bin. Everything looks pretty normal. Then we get to week 14. The soil looks like it's older. The bananas have decomposed. The chips have lots of mold on them, especially for the experimental bin. This is actually the tortilla chip right in here, if you can tell. The leaves have lost their color. So let's compare the leaves to week 1 bins. See, there are a lot vibrant colors in week 1 compared to week 14. The copy paper for the experimental bin has this it's kind of like a mold type wet spot on it almost. It became translucent. The chip bag, the Cheetos chip bag that I used in week 14, they became they're starting to fold in on each other. Nothing really happened with the screw. Nothing really major happened with the sticks. There became some green mold on it, and also this little white fungi type mold type of deal started to grow on it as well. So here's my data table for week 1. When I gathered these objects, I took down this information. So this is for my control bin, and this is for my experimental bin. Everything is relatively the same as my control bin, and then you'll notice when we get to the week 14 data table that a lot has changed. So here for data table 1, pretty much all of the objects are opaque, except for the bucket, which the bucket is like an ice cream bucket that I cut up, which it was translucent. And then we get into table 14, where objects became translucent, like the paper towel. For week 14, colors changed, weight changed a lot, size changed a lot. It was really interesting. So here's my control bin, and here is my experimental bin for week 14. You can also tell the difference between them because of the weights. So here the chip is 0.7 grams, and the chip in the experimental bin is 0.3 grams. So for data table 14, the stick is also really different than the controlled stick. So for the experimental stick, it is 1.7 grams, and then for the control bin stick, it's 2.5 grams. A little information that happened: the banana decomposed first in the experimental bin on week 8. The chips started to turn moldy in both bins on week 2, there were these little worm-like bugs that turned into little flying bugs. The longer that the experiment happened, the less bugs I saw. And then the fungi and mushroom type things started growing in week 2, but they were gone by week 8. They were gone in both bins. In my conclusion, overall my hypothesis was correct. The experimental banana did decompose faster than the control banana, and I also expected a lot more to happen. So that was kind of a shock that not as much happened, but still quite a bit happened actually. Things that I did wrong, I did not measure weight in week 1, so you notice my data tables look completely different for week 14 versus week 1 because I did not measure weight in week 1. And also, what went wrong, there was no consistent temperature. These bins were in my bedroom, so between week 1 and week 14, the temperatures fluctuated a lot in my room because when it's winter out, so when it's cold outside, it is like cold in my room. When it's hot outside, it's hot in my room. And since the bins were in my room, there's no consistent light. So it was nighttime and it was daytime. Improvements I would make, I would improve everything that I did wrong. I would try to make it right. And then also for my control bin, I would do nothing to it. And for my experimental bin, I would only do one thing instead of what I did, which was spray water on my control bin and spray water and vinegar on my experimental bin. It kind of defeats the purpose if you do something to your control bin. I should have just done nothing to it and then just did the vinegar for my experimental bin. But that is all I have. Thank you so much for listening, and it was a really fun project to do.